Hi, everyone. It's a pleasure to be here. And thanks for this opportunity to present my work. Today, I'm going to present my work on this disease, vector mosquito, Aedes aegypti. So what we are seeing here is basically a female Aedes aegypti mosquitoes that is feeding on human blood, and it needs human blood to reproduce. And during this process, it spread diseases like dengue, Zika, and yellow fever. So my work is basically to understand the molecular mechanism underlying this host-seeking behavior of these mosquitoes. And we can use this knowledge to basically then control this behavior and ultimately prevent the spread of the diseases caused by these mosquitoes. So the topic of my today's talk is channeling mosquito behavior by trip channels. So uh, I'm from Dr. Craig Montel's lab in UC Santa Barbara, and the main theme of our lab is to study sensory input and behavior in mosquitoes. And before diving into my work, I will just very briefly discuss about the trip channels. So trip channels are the sensors of temperature and natural products, and these are basically the conserved family of ion channels, which are found across many organisms from yeast to humans. These channels play a role in detecting wide range of environmental clues, including temperature, chemical signals, and mechanical stimuli. These channels are critical for sensory perception, and it allow organisms to respond appropriately to changes in their surroundings. Now, a unique feature about the strip channels is they note that these are the ion channels that can respond to completely different sensory modalities. Like the same channel can respond to the change in temperature, and the same channel can also respond to the natural products. So these are multimodal in nature. Like a same channel can respond to two totally different sensory modalities. And basically, this property of these channels will be the foundation of my work later. But let me go and understand why we want to study trip channels and why we want to study it in mosquitoes. So, as we know, mosquitoes are basically the deadliest animals, and the death toll due to these mosquitoes alone is more than the combined death toll of all the other animals that they cause. Two most deadly mosquito species are Aedes aegypti, which transmits dengue, Zika, yellow fever, and Anopheles, the primary vector of malaria. While Anopheles is responsible for a higher number of infections and deaths each year, which is particularly interesting about the Aedes is that it is far more invasive. It means it spreads rapidly to new regions, largely due to its remarkable ability to adapt to diverse and changing environments. Now, focusing on Aedes, it is predicted that global temperature rise and urbanization created more optimal conditions for the spread of this specific mosquito species, the Aedes, and this is a serious concern for the whole world. The recent trends of mosquito-based infections show a similar trend that although malaria cases and in deaths in the last decade have started to decline, the cases of dengue or the diseases that are caused by Aedes are rising sharply. So in our work, we try to understand the mechanism of host seeking in this specific mosquito species. Now, the ultimate goal of the mosquito research is to reduce the burden of mosquito-borne diseases. This can be approached in two ways. One is by controlling the mosquito population through methods like insecticides or genetic strategies such as sterile insect technique. However, these approaches come with concerns as mosquitoes play a very important role in the ecosystem. They are part of food chain and also serve as pollinator. So completely eliminating them could disrupt the natural ecological process. Another promising approach is to disrupt mosquitoes' ability to detect and bite humans. That's what we are focused on. Since only female mosquitoes bite to obtain blood meal necessary for the reproduction, preventing them from biting can effectively limit their ability to reproduce and reduce population growth over time. To achieve this, we need a very deep understanding of the sensory mechanism mosquitoes use to locate and bite humans, such as detecting body heat, carbon dioxide, skin odors, and then find the ways to interfere in these processes. To achieve this, it's very essential to deeply understand the sensory mechanism of mosquitoes, which they use to locate and bite humans. Why is it important to study multiple sensory cues and their underlying mechanisms? Is because female mosquitoes rely relies on the combination of signals such as volatile skin odors, carbon dioxide, vision, body heat, and humidity to find their host. Most of these cues, except for vision, are non-directional and can easily be diffused in the environment, especially in the presence of air movement or a moving host, making it very difficult for mosquitoes to determine the exact location of a person. However, mosquitoes' visual acuity is limited and only effective at short ranges. This raises an important question that what is the directional cue that guides mosquito towards their host from a distance? We explored this possibility that infrared sensing can be one of this sensory cue that provides directional information to the mosquitoes towards a specific host. Now, in the first part of my talk, I will discuss how mosquitoes use infrared sensing to narrow down onto their hosts. Now, once mosquitoes detect their host and then they basically decided to move towards the host, they began to explore the chemical composition on the surface of the host skin. And here comes the second behavior of the mosquito host seeking, that is post-landing behavior. Based on the chemical composition of the human skin, mosquitoes make decision whether they want to bite a person or not. So in the second part of my work, uh, I will investigate, or I will present how these gustatory sensing in the mosquitoes help them to understand how they make decision and how we might interfere with this characteristic of the mosquitoes to prevent them from biting humans. So coming to the first part of my work is I will discuss how Aedes mosquitoes use infrared radiation to detect and locate humans. Now, it all began with an interesting observation where we were actually trying to understand the thermal sensing or thermal stimuli, and in combination with other cues, how mosquitoes detect thermal signals from humans and human odor and CO2, and how these sensory signals are combined by mosquitoes to basically drive a host-seeking behavior. And during one of our experiments, we noticed that even the mosquito cage, when it is very far from the 34 degrees Celsius Peltier device, which we use to mimic the human skin, at even far enough distance, mosquitoes were able to basically go towards the side where there was a 34-degree Peltier. Now, the mosquitoes were still consistently landing on the side facing the heated Peltier, although the distance of the Peltier was very far from the cage of the mosquitoes. Now, when we measured the air temperature on both sides of the cage, we found that there is no difference in the air temperature, and the air temperature was uniform. So if conduction and convection are not reaching the mosquitoes, then there must be a third sensory cue, that is third component of the heat, that is called infrared, that mosquitoes must be sensing. And this led us to suspect that mosquitoes were detecting the infrared radiation that is emitted by the warm surface of the Peltier, and it was the only component of heat capable of reaching the cage from that far distance. So we started exploring this property. To test this idea that mosquitoes can detect IR, we modified our setup to put an additional layer of material and prevent any chance of convective heat reaching the cage. The IR source was at 34 degrees Celsius, which is an average skin temperature. We set this up in a way so that no conduction or convection heat can reach the mosquito cage. As a result, as shown in the figure, there was the two sides of the behavior setup, that is non-IR side and IR side, and there was no temperature difference on both the sides, but it was just the IR component of the heat that can basically reach the mosquitoes in this setup. So the temperature on both of the sides of the Peltier was uniform, and there was on the cage, and there was no temperature difference that the mosquitoes can sense. It was just the radiation component of the heat that the mosquitoes can sense. When we placed a mosquito cage with 80 female mosquitoes in front of the IR source, we found that there was a clear preference indexed towards the IR source as compared to the non-IR side on the mosquitoes. We ran the assay for 5 minutes, and during which we tracked mosquito activity, and we found that mosquitoes showed a clear preference for the IR emitting side of the cage. Interestingly, when we blocked the IR radiation from heated surface, this preference disappeared entirely. And these results indicate that IR can serve as a directional cue for Aedes aegypti during host-seeking behavior. So we concluded that IR may be a new human cue for Aedes aegypti mosquitoes, which drive the host-seeking behavior. So I want to highlight here that our method of tracking mosquitoes activity is different from many previous approaches, which typically focus only on the mosquitoes landing on the cage surface. Instead, we tracked a specific behavior associated with the activity of the host seeking, a probing behavior, and that mosquito exhibit when searching for a host, and incorporated this into our analysis. We calculated a preference index based on this behavior, and based on the difference in the number of these tracks observed on the IR side versus the non-IR side, we calculated the preference index in all our future experiments. And as you can see here, that the mosquitoes are showing very clear preference to the IR side versus the non-IR side. Now, next, we wanted to explore whether that IR synergizes with CO2 and human odor to drive mosquito attraction. Here, I'm showing that infrared is only an effective cue when it is combined with human odor and CO2 and not on its own. IR does not trigger host-seeking behavior itself. This makes sense because any object at 34 degrees Celsius wouldn't necessarily indicate a human host. So it's only the combination of sensory cues that drives mosquito behaviors towards the human host. This also helps explain why some earlier studies which investigate the role of IR in mosquito behavior may have failed to detect this important behavior, because they were looking at the role of infrared in isolation, but you will only get mosquitoes to show a preference towards IR in the presence of other important sensory cues like CO2 and human odor. We also found that IR is most effective as a sensory cue when it matches human skin temperature. In our experiments, we observed that as the Peltier surface temperature increased from 28 degrees Celsius to 37 degrees Celsius, the IR preference index also increased, peaking around 37 degrees Celsius. However, when we raised the temperature beyond physiological levels like 39 to 40 degrees Celsius, the response began to decline, indicating that the mosquitoes are tuned to detect IR within a specific biological relevant range. Additionally, IR is not only effective at a very short range, just a few centimeters when emitted from a small surface, but when we increase the size of the IR emitting surface to mimic the area of the human skin, we found that mosquitoes could respond to IR from a distance as far as the 70 centimeter. This finding is also critical in ruling out convective heat as a factor in our experiment, because convective heat from 34 degrees Celsius cannot reach such distances like 70 or 90 centimeter, which mosquitoes' response must be due to the infrared radiation component of the heat. So next, we propose what is the mechanism through which infrared radiation is detected, and how infrared-induced heating of. So we found that it is the infrared-induced heating of the fluid called endolymph in the specific structure called antenna that surrounds the dendrites extending from neurons at the end of the antenna. The mosquito antenna contains a lot of hairs called sensilla, including peg in pit sensilla at the very end of the antenna. The idea that the IR radiation would enter the aperture of the peg in pit sensilla, it bounces back, and the highly refractive internal housing heats up the endolymph in the sensilla. Instead, there is a neuron in this sensilla that is known to be activated by heat. So according to this model, the heat from our skin, which is converted to IR, is then converted back to heat, suggesting that the molecular sensor of IR radiation might be actually the infrared temperature sensor. So the molecular sensor that basically is detecting the infrared radiation is basically indirectly to the temperature sensing in the very tip of the antenna. However, so now moving to the mechanism of infrared sensing, we focused on TRPA1 channel, which has been previously identified as the thermosensor in Drosophila. In mosquitoes, few other labs like Hoshal group reported its role in thermal avoidance behavior. So we received these TRPA1 mutant lines from their lab, and interestingly, we found that when we tested these TRPA1 mutant mosquitoes in our IR preference index behavior, we found that TRPA1 mosquitoes showed no preference to IR, suggests that this channel is essential for IR detection. However, prior to our work, the expression and localization of TRPA1 in mosquito sensory organs has not been demonstrated. To address this, we performed in situ hybridization and discovered that TRPA1 is specifically expressed at the very tip of the antenna. Guided by this expression pattern, we dissected just the antenna tip of the mosquitoes, and we performed again the IR preference index behavior, and we found that removing only just the tip is sufficient to completely abolish the IR preference index in the mosquitoes, confirming that this region, that is the tip of the antenna, is critical for TRPA1 mediated IR sensing. Much of the genetic and sensory research in mosquitoes have traditionally been guided what is already known in Drosophila. And over the past decades, our lab, like Craig's lab, has uncovered unconventional role of Drosophila opsins, proteins traditionally recognized as photoreceptors. Our lab, along with others, have since demonstrated that opsins are just not only involved in vision, but also play important role in thermosensation, olfaction, and taste. In two independent studies, it has been shown that opsins in Drosophila play a role in thermosensory behavior, acting upstream of TRPA1. These studies suggest that opsins contribute to thermal signal amplification, enhancing flies' ability to detect temperature changes. So we wanted to explore if the similar mechanism also is playing role in Aedes and its mechanism in sensing infrared radiation or heat. So first, we explored which are the opsins that are expressed in Aedes, and we found that there are 10 opsins, and we identified the candidates potentially involved in infrared sensing. We first examined their relative expression in the antenna, which is the organ that is basically involved in IR sensing. We found that only two opsins, OP1 and OP2, are significantly expressed in the tissue. To test their functional relevance, we examined IR attraction in mutant line. Interestingly, we found that mutations in either OP1 and OP2 had no effect on IR attraction, but the double mutant show a significant reduction in IR preference, suggesting a redundant or cooperative role of both OP1 and OP2 in IR sensing. Next, we analyzed the expression of both these opsins, OP1 and OP2, in the antenna, and specifically in the region that is located at the tip of the antenna. And we found that both OP1 and OP2 are expressed at the very same location where we basically found the expression of TRPA1, that is a very tip of the antenna. So there is a possibility that both TRPA1 and these opsins are involved, or they interact with each other in sensing infrared radiation and deriving this infrared radiation preference index behavior. So our data indicate that opsins and TRPA1 function as heat sensor at the distal end of the antenna. To test this model, we performed electroantennograms, that is EAGs, using antenna exposed to IR from 34-degree sources. We found that at 34-degree stimulus, as compared to wild type, which elicits a normal response to the thermal or temperature sensory stimulus that is coming from the Peltier source. In contrast, in TRPA1 mosquitoes and opsin mutant mosquitoes, there is no signal in EAG to the thermal stimulus that is coming from the Peltier. This response elicited by the control was due to thermal IR, but there was no response in both TRPA1 and opsin mutant mosquitoes. So concluding, based on these observations, we made a conclusion, and our findings suggest that TRPA1 can directly detect high-intensity IR radiation. However, at low-intensity radiations, opsins play a critical role in significance amplification, enhancing the signal and ultimately, basically facilitating the activation of TRPA1. The role of opsin as amplification protein is conserved, much like what has been seen in Drosophila. And in the later part of my talk, I will show how the function is not limited to the thermosensory behavior in the mosquitoes, but the similar mechanism also, we observed the similar mechanism in chemosensory behavior of the mosquitoes. So I will come to the second part of my talk, that is the post-landing behavior. So in the first part of my talk, I discussed how opsins and TRPA1 interact with each other to derive the thermosensory or infrared sensory behavior of the mosquitoes. And in the second part of my talk, I will discuss how same proteins through a similar mechanism is driving a completely different sensory behavior, that is chemosensory behavior, different from the thermosensory behavior. So in our lab, in Craig's lab, we have shown that in Drosophila, that certain plant-derived phytochemicals are aversive to flies, and this response is mediated by opsins that are expressed in the fly taste system. This aligns with a common strategy in nature because plants produce these phytochemicals as a defense mechanism to protect themselves from insects and the pests. So many of these chemicals functions as toxin as they are bitter chemicals or disrupt the insect sensory pathways. Our goal is to basically use this natural strategy to identify phytochemicals that can basically activate mosquito opsins and in doing so trigger aversive response to prevent mosquito biting. So we want to identify if the similar mechanism of sensing plant-derived chemicals is there in the mosquito Aedes aegypti, and can we use this approach to basically identify new chemicals that can later be used as potential repellents in the mosquitoes. So do Aedes use opsins as taste sensors? So first, we examine the relative expression of mosquito opsins in taste organs in Aedes mosquitoes, specifically the labellum and the tarsi. Interestingly, we found that the same two opsins that were highly expressed in the antenna of the mosquitoes also show a very strong expression in both the labellum and all the tarsi. The foreleg, the mid legs, and the hind legs, only the two opsins are mainly expressed. That is the same opsins, OP1 and OP2. And the expression of these opsins is not just few folds high, it is like 100 or 1,000-fold higher than the other opsins that are expressed in these tissues. Now, to validate their expression, we visualized their localization and performed in situ hybridizations, and we found that, in fact, both the OP1 and OP2 are expressed in the labellum as well as the forelegs and the hind legs and mid legs as well, which I'm not showing in this figure. But these two opsins are the mainly expressed opsins in these taste organs of the mosquitoes. Based on this strong expression in gustatory tissue, we selected these two abundant opsins and expressed them ectopically in HEK cells, and we screened these opsins with a library or a small library of the bitter compounds, that is 128 natural bitter compounds, to basically identify any specific bitter chemical that can activate these opsins. And we found that a specific chemical, that is a flavanol called catechin, is activating both OP1 and OP2. And both catechin is basically a bitter flavonoid that belongs to a flavanol family, and it is found at particularly high concentration in some natural plant products like green tea, and it's also present is very less amount in some fruits, but it's a natural bitter chemical which is basically activating both OP1 OP2 in our in vitro screening. Now we wanted to test how mosquitoes respond to catechin in behavior assays. So we designed this two-way behavior assay. In this setup, we placed 40 female mosquitoes in the cage and offered them a choice between two blood feeders. One is basically coated with the water as a control, and the other is coated with varying concentrations of catechin. To score the mosquitoes, we add a fluorescent dye in the blood to score them after the feeding assay is done. And we found that the mosquito began avoiding catechin at very relatively low concentrations, specifically beyond 2.5 to 5 millimolar concentration. Mosquito was totally avoiding the blood, which was basically towards the catechin side. While it may seem like interesting that, okay, this is a bitter chemical, so mosquitoes are avoiding bitter chemicals, so what's interesting about it? So we tested other bitter chemicals, which are basically the known bitter chemicals, not even for the humans, but also for the flies, and we tested them in the similar assay. We also take some standard flavanols, which are structurally similar to catechin, like epicatechin, EGCG, and genistein, but we also taken some very bitter chemicals like quinine, caffeine, and naringin, which are standard bitter chemicals. And we found that mosquitoes are not avoiding these standard bitter chemicals, quinine, naringin, caffeine, but they are only avoiding this specific class of bitter chemicals that are structurally related to each other, that is structurally related to catechin. Now, we also, other than this, we also performed egg laying assays where we basically put two egg laying cups, one with water and one with 10 millimolar catechin, and we allow the mosquitoes which were blood fed to lay eggs in these cups. And we found that as compared to water, where the egg laying was very high, very few mosquitoes lay eggs, or there were very few eggs in the catechin coated cups or the cups which have 10 millimolar catechin in them. So from these experiments, we conclude that, okay, it's mosquitoes are avoiding catechin, and it's not general avoidance to all the bitter chemicals. It's avoidance to a specific, this chemical, catechin, or a similar class of chemicals that are structurally related to this chemical. Now, to further investigate the underlying mechanism of this catechin aversion, we conducted single blood choice behavior assays where we take wild type control mosquitoes and we perform single choice blood feeding behaviors in this mosquito. And here, as you can see, that mosquitoes are in fact uh, going towards the feeders where there is just a control water, and they are feeding and sitting there, and they are basically the wild type mosquitoes, in fact. Uh, are basically avoiding sitting on the catechin coated cups. And this is basically the 5 millimolar catechin video, which I'm showing. In fact, there is a blood feeder there, but mosquitoes are sitting there, making a contact with the membrane, and they are flying away. Now we tested OP1, OP2, as well as TP1 in the same behavior assay, and we found that as compared to at the same concentration, of catechin, as you can see that OP1, OP2 mosquitoes are landing and they are trying to blood feed. But if you look at the TP1 mosquitoes, they are not avoiding even sitting on the surface and blood feeding, and mostly, like almost all of them, are going towards the blood feeding membrane and they are trying to blood feed. As compared to that, if you look at the wild type, there is very clear avoidance to the catechin coated membrane feeding surface. So TP1 basically mosquitoes are showing rescue or are showing defect in sensing catechin. This defect is there in OP1, OP2 as well, but the defect is not as severe as it is in the TP1 mosquitoes. So TP1 mosquitoes, as you can see here, are clearly not able to sense catechin as compared to the wild type mosquitoes. So from these behavior assays, we conclude that TP1 has a role in sensing catechin, but OP1, OP2 is also involved in this sensing mechanism. So we haven't basically looked at the expression of TP1 in mosquito taste organs, specifically labellum. So we generated a new TP1 reporter line, TP1 QF2, that is basically used to drive the expression of MCDH GFP in the taste structures of the mosquito, that is proboscis and the legs. And we express basically performed immunostaining, and we detect basically TRPA1 labeled neurons in both the labellum as well as foreleg, midleg, and hind leg. So TRPA1 is expressed in all the sensory structures that are involved in sensing or that are involved in chemosensory behavior. Next, we use the same line to basically use the calcium erasing in live mosquitoes when they were exposed to catechin. So the QF2 line was crossed with GCAM6, and we exposed these mosquitoes to the 10 millimolar catechin concentration. And as you can see that not all, but a specific subset of TP1 neurons were activated by 10 millimolar catechin in live calcium imaging in both labellum as well as tarsi. So TP1 is basically sensing catechin directly, and this is the mechanism how basically mosquitoes are showing aversion to catechin, and that's why TP1 mutant mosquitoes are not able to sense catechin. To show it directly, we also performed basically the recordings from the gustatory receptor neurons. This is basically the tip recording from the extracellular tip recordings from labeller taste sensilla. And based on the expression, we perform these recordings from the specific hair, which is basically where we can see the expression of TRPA1. And we found that the loss of OP1 and OP2 as well as TRPA1, the response to the catechin in the wild type is basically diminished in OP1, OP2 double mutant, but this response to catechin is totally gone in TRPA1 mutant lines, that is two independent lines that we have analyzed for our behavior. So this is the direct evidence that both these proteins, opsins as well as TRPA1, are involved in sensing catechin, and loss of these proteins from mosquito gustatory structures basically lead to loss of their sensitivity to this bitter chemical catechin. Now, we know that TRPA1 is involved in sensing catechin, but it's known that different isoforms of TRPA1 have different functions, and that may be the reason that TRPA1 same channel is involved in sensing two different sensory modalities, thermosensation as well as chemosensation. So we basically try to explore what are the specific isoforms of TRPA1 which are involved in chemosensory mechanism. So we performed PCRs by isolating RNA from the labellum as well as the forelegs, mid legs, hind legs, and we performed isoform-specific PCRs for these different isoforms, and we found that a specific isoform called TRPA1G is mainly expressed in all the organs, the proboscis, foreleg, mid legs, and at very less amount to hind legs, which makes sense because hind legs are the only tarsi which do not make contact when mosquitoes basically are sitting on the surface of the of blood feeding surface like membrane or the human skin. It's the proboscis, forelegs, and mid legs which are making the first contact with the feeding surface, and hind legs do not make contact. So that's the reason maybe that the chemosensory isoform, that is TRPA1G, is only expressed at very high levels in only these structures. So next we decided to basically test these different isoforms in electrophysiological assay, where we basically expressed TRPA1G in xenopasteurocytes and performed voltage clamp recordings following catechin application. We found that not only just in Aedes, in Anopheles, TRPA1G is activated by catechin, and we take AITC, that is known TRPA1 activating, as a positive control, and we found that in both Aedes aegypti TRPA1G and Anopheles TRPA1G are activated by catechin as well as AITC. Now, another interesting thing to test is the obvious choice is to basically test the human TRPA1, because if human TRPA1 is also activated by catechin, this chemical will be irritant to human skin, and there is no way to basically use this chemical as a possible future repellent, because any possible repellent should not activate human TRPA1, because then it will be a natural irritant to human skin. So we tested human TRPA1 in the similar xenopus oocyte recordings, and we found that, in fact, AITC, which is the positive control, is activating human TRPA1, but there is no activation of human TRPA1 by 1 and 5 millimolar catechin. So based on these observations, we concluded that the response of TRPA1 to catechin is conserved in insects. We later also checked the Drosophila TRPA1, and we found that even Drosophila TRPA1 is also activated by catechin, and human TRPA1 is not activated by tryptophan. So this response to catechin is insect specific, but as we move from insects to higher organisms like humans, the TRPA1 is not activated by catechin. So based on these observations, we found that there is a potential to basically modify this chemical as a possible repellent or contact-based repellent, because this chemical is specific to insects, and it do not activate human TRPA1. Based on these observations, we concluded that the mechanism of chemical activation of TRPA1 mirrors what we observed in our thermosensation studies. TRPA1 can be directly activated by high levels of stimulus, but at low levels, in both cases, thermosensory behavior and chemosensory behavior, the low concentration of chemical or the low intensity of radiation is activating auxins, which in turn is amplifying this signal and indirectly is activating TRPA1. So both pathways ultimately converge and activate TRPA1, which is the final player in this mechanism, leading to strong aversive response in mosquitoes in case of catechin. But in case of infrared radiation, the TRPA1 might be expressing in not in the aversive neurons, but the attractive neuron. So it's driving attractive behavior. So both the mechanisms, both the sensory modalities have conserved mechanism where opsins are basically driving TRPA1. But in this case, this behavior is driving aversive behavior. That's why mosquitoes are showing aversion to catechin in this case. So finally, I would like to basically thank all the current and former lab members in the CREGS lab. So this is Nick, who collaborated with me in the IR project, and Pratik, who collaborated with me in the catechin product. We have submitted the second catechin's story, and I would be happy to take any questions based on my own.
Right, thank you so much, Avanish, for that really fascinating talk. So as usual, we'll take questions in the chat, or if you would like to ask your question directly, please raise your hand, and then Greg will make you a panelist, and you'll be able to talk directly to Avanish. All right. While we're waiting for our first question, Avinash, I was wondering, have you looked at what kind of subtype of A1 is expressed in the antenna?
Yes. So because when we were doing basically the IR study like 3 years back,
Mhm.
we don't have tools. We didn't. So we generated one QF2 line recently for our second project. So at that time, we were not able to go into the isoform specificity in that
Mhm.
behavior. But now, even after publishing, we tested, and it was basically the one A isoform, which is basically a thermosensory isoform, which is expressed specifically in the antenna. But the isoform that is expressed in the proboscis and the tarsi is a different isoform, that is one G isoform, that is basically a chemosensory isoform. So the
Yeah,
nature of the isoforms that express in both the structure decides what kind of sensory modality they are going to derive. Yeah,
that's fascinating. And so the
yeah.
the subtype in the antenna is not as sensitive to catechin.
No, no, not
Yeah,
sensitive to
not
question.
sensitive at
Yeah,
all. That's
no, no,
fascinating.
yeah. So,
We have a raised hand to Soon-Ju
Oh yeah,
Lee.
here we are Q&A. We have Maurice Cannon asked, do catechins and IR act synergistically in any behavior or physiology?
okay. We haven't tested this, but it will be a little different because both of these are driving opposite sensory modalities. Catechin is deriving an aversive sensory modality because it is expressed in the gustatory tissues in the bitter neurons or aversive neurons, and infra and one that is a different isoform. I said in the antenna is basically expressed in the attractive neurons that are deriving attraction towards the thermal preference. So we have not tested because this is like a isoform specific behavior, but we haven't tested. I understand like the IR preference behavior in presence of catechin, so we have never done that. So they may like contradict each other because they both are two different sensory modalities interfering with each other. Yeah.
Right. So Sanjay Lee asks, is TRPA1 type G not activated by heat?
Triple one, which gene? Sorry.
Type G, so isoform
Okay.
G.
One type G is activated by thermal sensor. We have done Xenopus experiments, but the activation is not as strong as other isoforms one. So one, I won't say that it's not activated by temperature. It's activated by temperature, but this is the only isoform that is activated by chemical. The other one isoforms are not activated by chemical. So it's basically most of the one isoforms can be activated by temperature, but this is the only isoform that is activated by chemicals. So this is activated by temperature, but at a very less levels as compared to the other isoforms, which are standard thermal activated one isoforms. Yeah.
Right. Damien Bell asks, very impressive work, Dr. Chandel. Thank you and congratulations. An obvious question, have you humanized TRPA1G by taking human sequence differences and adding to make it catechin insensitive, and vice versa for human A1 to make it sensitive to catechin by adding TRPA1G sequences?
We haven't done that. That's an interesting question, actually, to basically change the human one isoform to mimic basically the mosquito one isoform. We haven't done that because there are, although they are conserved proteins, but the sequence similarity is not that high. And there are very critical differences at the N and C terminal, but we haven't done this experiment. We have never basically tried to change human one into insect and see if started basically showing our shell response or to catching. We haven't done that, but that's an interesting experiment to do. Yeah.
Here's another question from Maurice Cannon, who asks, as a follow-up to my earlier question, would you expect antagonistic or synergistic action at a single cell level of heat and catechins, I imagine?
Because these are different isoforms, there will not be a synergistic action because they are driving two different, as I said, they are driving two different modalities. One is driving aversion and one is driving attraction. So they are going to antagonistic action. They are going to basically contradict each other because two different sensory modalities which are driving two different type of behaviors. They are not going to synergize each other. The catechin is driving aversive role, and the infrared is deriving attractive role. So they are going to contradict each other. So we cannot predict which is going to basically uh, win over each other or dominate over each other, but they are not going to synergize. They are going to antagonize each other. Yeah.
Oh, I should also mention that, Marie says, thanks, cool work. I
Okay.
should also mention to anyone that that's in attendance that might be on a search committee that Avinash is on the job market currently, so keep an eye out for his application.
Okay.
Okay, here's another question from Damien Bell. You alluded to this in the catechin studies. I guess catechin would make a great repellent.
That's actually an idea. The only issue with the plant products is that, unlike the chemical repellents, there is a factor called of stability. The plants continuously produce these chemicals; they don't store them. So, anytime we isolate a plant product and we want to basically uh, propose it as a repellent, we have to look at the factor how stable it is. And the half-life of the catechin, when we perform these experiments, is not that high, and within few hours it starts degrading, so the efficiency starts there. So what we are trying to basically now do is trying to find the analogs or modify catechin and make a formulation of catechin in such a way so that we can increase the stability of this chemical, like few days, weeks, or maybe like the months. So in that case, basically, yes, it's a very good potential uh, uh, repellent, but we have to figure out how to basically increase the stability of it so that it can last long, and it can compete with the chemical repellents which are already out there, like DEET and other chemical repellents. Yeah.
Damien Bell says, thank you again, great work.
Thank you.
Right. Okay, so here's a question for Pablo Gaete. I wonder if this mechanism is translated to other mosquito species.
So we have tested, although I just showed the electrophysiological experiments with Anopheles, but we have performed behavior assays with both Aedes and Anopheles, and we found that both the species show aversion to catechin at a very low concentrations. We haven't tested other species like Culex, but but if we just take the TRPA1 isoforms from those species and test them electrophysiologically, and they show the activation by catechin, then we are pretty sure that behaviorally also they will show the aversive response to catechin. So the simple experiment to do is just take the TRPA1 from these specific insect species and test them in electrophysiology experiments, and we're pretty sure that it will be reflected in behavior assays.
Avinash, I don't remember if in your talk, if you showed this, what is the chemical structure of catechins?
I have shown it. Let me
Yeah,
go.
you
I don't know.
probably did. I just don't
Yeah,
remember.
I don't know, but yeah, it's a cyclic, three cyclic rings. Let me go. Yeah, this is the structure.
Oh,
Yeah.
okay. Okay.
Yes. Yes.
Okay. So have you looked at like chemicals that are related?
So that's
Oh,
the
I
one
guess other flavonoids.
we did
Yeah.
experiments where we basically take the chemicals which are structurally similar to this chemical, and we found that the chemicals which are like from the same class and structurally pretty similar, they also show the similar behavior, not to the same extent as catechin, little less, but they show the similar behavior. On the other side, because it's a bitter chemicals, so we take some standard bitter chemicals like quinine, denatonium, which are like the most bitter chemicals
Yeah.
known to humans. But mosquitoes do not show aversion to those bitter chemicals. So it's not related to bitterness. It's basically
It's the
related to their
structure.
ability to
Yeah.
activate TRPA1. Yes. Yeah.
Yeah. Yeah. Okay. All right, so here's an anonymous attendee who asks, have you found sex age-dependent changes in catechin sensitivity or TRPA1 expression in mosquitoes, I'm guessing?
We have tested males. So males do not show aversion to catechin. Makes sense because males do not blood feed, so they do not need TRPA1 in their sensory structures. So we have found that the aversion males show is at very high concentration that are not basically as close to the females. So yes, there is a sex factor, but there is no age factor. We have tested. Yes, we don't test too young mosquitoes because TRPA1, these proteins need some time during development to basically have enough expression in the sensory structures. But once we get to the matured adults, we see a consistent response to catechin throughout the age. So yes, there is a sex factor, but not age factor to the sensitivity of the catechin. Yes.
From Pashu Sunalane.
Yeah, that's very interesting. Yeah, I read that. Yeah, I don't know how much by drinking because catechin is just a very fraction of the green tea. Although we know that our eating habits, if people who drink alcohol are more bitten by the mosquitoes because their skin composition changes and there is more content of ethanol in their sweat, so they are more attracted to mosquitoes they are more driven to attack by the mosquitoes, but not sure about the green tea because we don't know how much is the effective concentration that will basically reach the skin, and is that enough to basically repel mosquitoes, and how the catechin is metabolized inside our system. So that is something interesting, but yeah, it will be a little tough to do that kind of experiments. Yeah, but that's an interesting insight. Yeah.
Maybe a cream made of green tea. Excellent.
Yes, something like that. Maybe in the moisturizer or something like that. So that can be something, but I don't think drinking would be a solution. Yeah.
Yeah, speaking of like things that there you go. It looks like you won't even need to be on the job market, Avinash. You're straight
Yes,
into
yeah. I think that was
a startup.
yes.
All right. Well, if there are no more questions, then I guess we'll wrap up.
Thank you.